Mitosis consists of 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. It is preceded by a process of cell growth known as interphase, in which there are subphases: G1 phase, S phase, and the G2 phase. In the G1 phase, the cell grows. In the S phase, the cell continues to grow and duplicates its chromosomes. And in the G2 phase, the cell grows and prepares for the mitotic or M phase, in which the cell will divide. Specifically, in the G2 phase of interphase, the nuclear envelope encloses the nucleus of the cell, and the centrosome duplicates to form 2 centrosomes. These centrosomes located in animal cells organize the microtubules of the spindle and contain 2 centrioles. Additionally, chromosomes cannot be seen after the duplication in the S phase yet because they still remain uncondensed. Ideally, after the G2 phase, the cell will enter the M phase, entering the subphase called prophase. In prophase, the chromatin condenses, resulting in distinctly visible chromosomes. Each of the duplicated chromosomes is displayed as 2 identical sister chromatids connected at the centromeres. Additionally, the nucleoli disappear, and the mitotic spindle, composed of centrosomes and microtubules, starts forming. Asters can be identified in prophase as well. There are radial arrays of microtubules that extend from the centrosomes, which start to move away from each other as the length of the microtubules increases.
Because they made the membrane.
The cell then enters prometaphase, where the nuclear envelope fragments and microtubules extend into the nuclear area.
They do, they don't.
How are they showing up?
They just took it off.
Each chromatid of the chromosome has a protein structure in the centromere called a kinetochore. Microtubules attach to these kinetochores, acquiring the name kinetochore microtubules, and they move the chromosomes back and forth.
Okay, so
Do you want me to start?
that's what I said.
They're doing this, so they have this, and then they take it away, and then they throw like fragments onto the paper.
That's a good idea.
So we already have fragments, so we can just take this out and then just throw like those pieces of paper onto the.
You work one of those.
We don't need to. We already have. We literally already have pieces.
And maybe we could.
The non-kinetochore microtubules interact with the non-kinetochore microtubules on the opposite pole. The cell division transitions into the metaphase, which is the longest stage, about 20 minutes of mitosis. The centrosomes are at the opposite ends of the cell, and the chromosomes are lined up at the metaphase plate, which is an imaginary plate between the spindle's 2 poles. The kinetochores of the sister chromatids of each chromosome are attached to kinetochore microtubules from opposite poles. The entire apparatus of microtubules is known as the spindle due to its shape. The stage preceding metaphase is anaphase, which in contrast is the shortest stage, and this is when the sister chromatids separate and become individual chromosomes. The kinetochore microtubules shorten as the 2 parted chromosomes begin moving towards opposite ends of the cell.
So at this point, they have lenses, so this is gone, they're fragments. Just throw fragments in, like
We need to be on
this.
the right way.
Due to the microtubule being attached at the centromere region, the chromosomes move centromere first. The elongating cell has 2 ends that each have an equal number of
Do you want
chromosomes.
to practice some?
The final stage of mitosis is telophase, and the final stage of the M mitotic phase is cytokinesis. In telophase, 2 daughter nuclei, which are genetically identical, begin to form, and nuclear envelopes form from fragments of the parent cell's nuclear envelope and additional parts of the endomembrane system.
You don't need a
We can
chromosomes.
keep literally all the ones that are on here. That's all we need.
The chromosomes become less condensed in mitosis or nuclear division ends, while in telophase the division of cytoplasm begins, but the process is completed in a preceding stage called cytokinesis. The cleavage furrow, which pinches the cell in 2, forms in animal cells, and the cell plate forms in plant cells during cytokinesis as well.